Hello viewers, today I will discuss about TCA cycle and ETS. TCA cycle is tricarboxylic acid cycle and ETS is electron transport system or electron transport chain. Oxidation of glucose. The complete oxidation of glucose proceeds in different stages including number 1 glycolysis, pyruvate oxidation, TCA cycle, electron transport chain and chemiosmosis. TCA cycle. It is also known as Krebs cycle because it was discovered by Hans Krebs and William Arthur Johnson in 1937. TCA cycle essentially involves the oxidation of acetyl-CoA to carbon dioxide and water. It is the central metabolic pathway. The cycle is the final common oxidative pathway for carbohydrates, fats, amino acids. TCA cycle supplies energy and also provides many intermediates which are required for the synthesis of amino acids, glucose, heme porphyrin, etc. The cycle is the most important central pathway connecting almost all the individual metabolic pathways. The purpose of the TCA cycle is to convert acetyl-CoA to carbon dioxide to generate reducing equivalents NADH, FADH2, which in turn are oxidized in the respiratory chain to generate ATP as well as GTP generation. Now this acetyl-CoA, it comes from the pyruvate from the glycolysis of glucose, from fatty acids, from ketogenic amino acids. These acetyl-CoA can be served for oxidation of many metabolites, fatty acid synthesis, for cholesterol biosynthesis, steroid hormone synthesis as well as to generate ketone bodies. Now, the TCA cycle is a series of enzyme catalyzed chemical reactions having the central importance as a part of cellular respiration. The citric acid cycle begins with the transfer of a 2-carbon acetyl group from acetyl-CoA to the 4-carbon acceptor compound oxaloacetate to form a 6-carbon compound citrate. Most of the energy made available by the oxidative steps of the cycle where from NAD+, NADH is getting generated which goes inside the oxidative phosphorylation or electron transport chain. FADH2 is covalently attached to succinate dehydrogenase, an enzyme functioning both in the TCA cycle and the mitochondrial electron transport chain. This TCA cycle is an open cycle and aerobic in nature. The name is sometimes called citrate or citric acid cycle because citrate is the first product in the cycle. The location of TCA cycle. The reaction occurs in mitochondrial matrix in close proximity to the electron transport chain in case of eukaryotes. In prokaryotes, in the cytosol with the proton gradient for ATP production being across the plasma membrane happens. Reaction of TCA cycle. So, what I have already told, acetyl-CoA will condense with another metabolite, will produce the citrate. Now, this acetyl-CoA will be generated from pyruvate via pyruvate dehydrogenase complex. So, oxidative decarboxylation of pyruvate to acetyl-CoA will be done by pyruvate dehydrogenase complex. So, this reaction, that is the conversion of pyruvate to acetyl-CoA, is a junction between glycolysis and TCA cycle. It happens via a multi-enzyme complex, that is pyruvate dehydrogenase complex. What happens here? Pyruvate's carboxyl group, which is fully oxidized and is removed to release carbon dioxide. The remaining 2 carbon is oxidized and form a compound named acetate. A particular enzyme transfers the extracted electrons to NAD+ and NADH will form. Finally, coenzyme A is attached to the acetate by an unstable bond and this makes acetyl group very active. Okay? So, acetyl-CoA has a high potential energy and will undergo inside the citric acid cycle and ultimately release energy to make ATP. So, pyruvate dehydrogenase is a multi-enzyme complex which converts pyruvate to acetyl-CoA. There are 3 enzymes are present. Number 1, pyruvate dehydrogenase. Number 2, dehydrolipoamide acyl transferase. And number 3, dehydrolipoyl dehydrogenase. The reactions of TCA cycle. Step 1, formation of citrate. Oxaloacetate condenses with acetyl-CoA to form citrate, which is catalyzed by the enzyme citrate synthase that is inhibited by ATP, NADH. Citrate is competitive inhibitor of oxaloacetate. Step 2 and 3. Citrate is isomerized to isocitrate. So when this isomerization happens, it happens by the help of enzyme aconitase. This is achieved in a two-stage reaction of dehydration followed by hydration through the formation of an intermediate called cis-aconitate. Steps 4 and 5. Formation of alpha-ketoglutarate. Isocitrate dehydrogenase catalyzes the conversion of or oxidative decarboxylation of isocitrate to oxalosuccinate and then alpha-ketoglutarate. The formation of NADH and the liberation of carbon dioxide occur at this stage. Now, stimulated in the cooperative way, this enzyme is by the isocitrate, NAD+, magnesium 2+, ADP, calcium, which is actually linked with the concentration. And the enzyme is inhibited by NADH and ATP. Step 6. Conversion of alpha-ketoglutarate to Succinyl-CoA occurs through oxidative decarboxylation. It is catalyzed by alpha-ketoglutarate dehydrogenase complex. So, alpha-ketoglutarate dehydrogenase once again is a multi-enzyme complex. At this stage of TCA cycle, second NADH is produced and the second carbon dioxide is liberated. Step 7. Formation of succinate. Succinyl-CoA is converted to succinate by the help of enzyme Succinyl-CoA synthetase. This reaction is coupled with the phosphorylation of GDP to GTP. This is a substrate level phosphorylation. GTP is converted to ATP by the enzyme nucleoside diphosphate kinase. Step 8. Conversion of succinate to fumarate. Succinate is oxidized by succinate dehydrogenase to fumarate. This reaction results in the production of FADH2. Step 9. Formation of malate. The enzyme fumarase catalyzes the conversion of fumarate to malate with the addition of water. Step 10. Conversion of malate to oxaloacetate. Malate is then oxidized to oxaloacetate by malate dehydrogenase. The third and final synthesis of NADH occurs at this stage. The oxaloacetate is regenerated, which can once again combine with another molecule of acetyl-CoA and can continue the cycle. So, count of carbons. 3 carbon pyruvate generates 2 carbon acetyl-CoA. Along with 4 carbon compound, it will generate 6 carbon citrate. Then it will be converted to another 6 carbon compound. By liberation of 1 carbon dioxide, it will generate 5 carbon compound. Once again, by liberation of another carbon dioxide, will generate 4 carbon compound, which can be converted to another 4 carbon, which can once again be converted to another 4 carbon, and this is converted to another 4 carbon compound, which lastly can give rise to 4 carbon oxaloacetate. So, in this way, actually this happens twice for each glucose molecule. Regeneration of oxaloacetate. The TCA cycle basically involves the oxidation of acetyl-CoA to carbon dioxide with simultaneous regeneration of oxaloacetate. There is no net consumption of oxaloacetate or any other intermediate in the cycle. Significance of TCA cycle: complete oxidation of acetyl-CoA, ATP generation via electron transport chain, final common oxidative pathway, integration of major metabolic pathways, fat is burned on the wick of carbohydrates, excess carbohydrates are converted to neutral fat, no net synthesis of carbohydrates from fat, carbon skeleton of amino acids finally enter the TCA cycle. Requirement of oxygen by TCA cycle. There is no direct participation of oxygen in the cycle. It operates only under aerobic conditions. This is due to NAD plus FAD required for the operation of the cycle can be regenerated in the respiratory chain only in the presence of oxygen. Therefore, citric acid cycle is strictly aerobic. The regulation of TCA cycle. The cycle can be regulated by allosteric regulation of metabolites. Indicator molecules of higher energy state, that is ATP, NADH, citrate, acetyl-CoA, they inhibit TCA cycle. But the indicator molecules of low energy state, that is ADP, AMP, NAD+, that stimulate TCA cycles. Citrate is used for feedback inhibition. It can be regulated by calcium as well as transcriptional regulation is there. There are 3 allosteric enzymes. So, all those enzymes are regulatory enzymes which can regulate TCA cycles. The enzymes are citrate synthase, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. Citrate synthase is inhibited by ATP, NADH, Acetyl-CoA and Succinyl-CoA. Isocitrate dehydrogenase is activated by ADP and inhibited by ATP and NADH. Alpha-ketoglutarate dehydrogenase is inhibited by Succinyl-CoA and NADH. Availability of ADP is very important for TCA cycle to proceed. Inhibitors of TCA cycle. Aconitase enzyme is inhibited by fluoroacetate, which is non-competitive inhibitor. Alpha-ketoglutarate is inhibited by arsenite. This is once again also a non-competitive inhibitor. Succinate dehydrogenase is inhibited by malonate, which is competitive inhibitor. Amphibolic nature of TCA cycle. TCA cycle is both catabolic and anabolic in nature, called amphibolic. Since various compounds enter into or leave from the TCA cycle, it is sometimes called as metabolic traffic circle. Important anabolic reactions of TCA cycle. Oxaloacetate is precursor for aspartate. Alpha-ketoglutarate can be transaminated to glutamate. Succinyl-CoA is used for synthesis of heme porphyrin. Mitochondrial citrate is transported to cytoplasm and it is cleaved into acetyl-CoA to provide substrate for fatty acid synthesis. Anaplerosis or anaplerotic reactions. The reaction concerned to replenish or to fill up the intermediates of TCA cycle are called anaplerotic reactions or anaplerosis. Important anaplerotic reactions. Pyruvate carboxylase catalyzes the conversion of pyruvate to oxaloacetate. Then by transaminases, conversion from glutamate to alpha-ketoglutarate. Once again from aspartate to oxaloacetate by another transaminase, from pyruvate to malate. The metabolic defects, extremely rare but it happens. Defects in pyruvate dehydrogenase enzyme complex causes lactic acidosis, neurological disorders. Defect in pyruvate carboxylase can cause hyperammonemia as well as lactic acidosis. Variation. So, some of the enzymes are varied in different archaebacteria like Acetobacter aceti. In spite of isocitrate to alpha-ketoglutarate conversion enzyme, here Succinyl-CoA to acetate-CoA transferase is used, and in Helicobacter pylori, the enzyme used is Succinyl-CoA is to acetoacetate-CoA transferase. Energetics of citric acid cycle. Now, how many ATPs are synthesized here? From isocitrate to alpha-ketoglutarate, 1 NADH is produced, that is equivalent to 3 ATP, or maybe by current research 2.5 ATP. From alpha-ketoglutarate to Succinyl-CoA, once again 1 NADH, maybe 3 ATP equivalent or 2.5 ATP. Succinyl-CoA to succinate, GTP, that is 1 ATP equivalent. Succinate to fumarate, FADH2, that is equivalent with either 2 ATP or 1.5 ATP. Then malate to oxaloacetate, once again another NADH, it may be 3 ATP or 2.5 ATPs. So, either as a total 12 or 10 ATPs are produced. Now, net ATP production depends on the shuttle used for the transfer of reducing equivalents from cytosol to mitochondria. So ATP generation during oxidation of glucose by glycolysis, number of ATP per molecule of glucose may be either 8 or 7 by new concept. Pyruvate dehydrogenase complex either 6 or 5. Citric acid cycle 24 or 20. So the total may be 38 or other by new concept 32. Electron transport system or electron transport chain. Chemiosmotic mechanism of oxidative phosphorylation. Active transport carrier proteins set up gradients which are then used to synthesize ATP by ATP synthase. Electron transport chains are the redox reactions that transfer electrons from an electron donor to the electron acceptor. So it's a kind of redox system. So lowest redox potential happens within NADH. Okay, that means it will transfer electron to FMN, to the next acceptor. The transfer of electron is coupled to the translocation of protons across a membrane, producing a proton gradient. The proton gradient is used to produce work, maybe in the form of ATP or others. Electron transport and ATP generation location. The enzymes and electron carriers for electron transport are located along the inner membrane of the mitochondria in eukaryotic cells. The reduced coenzymes like NADH and FADH2 produced from glycolysis, oxidation of pyruvate and citric acid cycle are oxidized to provide energy for the synthesis of ATP. The electrons are quickly passed along the respiratory chain to molecular oxygen to form water. Energy released during the passage of electron is used to pump protons and create a proton electrochemical gradient. The proton gradient drives the synthesis of ATP by oxidative phosphorylation. The addition of phosphate groups to ADP using this process requires oxygen. These events also take place in aerobic bacteria using plasma membrane. Electron transport system. The electron transport system, there are 5 protein complexes which are numbered as 1, 2, 3, 4 and 5. Each complex contains metal ions that form a pathway for the passage of electrons through the complexes. This powers the pumping of protons. 2 electron carriers are there, coenzyme Q and cytochrome C, attached to the inner membrane of the mitochondria. So they carry electrons between these protein complexes bound to the inner membrane. In electron transport, the oxidation of NADH and FADH2 provides the hydrogen ions and electrons that eventually react with the oxygen to form water. The linking of electron transport, proton pumping and ATP synthesis is called chemiosmotic coupling. The protons are positively charged. They will move across the membrane. It has an excess of negative electrical charges on the other side. Therefore, a proton gradient created across the inner mitochondrial membrane and is very steep, consisting of the electroattractive force and the concentration gradient set up by pumping protons into the intermembrane space, which can cause to make electrochemical gradient. This gradient is used to drive ATP synthesis by oxidative phosphorylation. So I have told you about different complexes. Let's start with complex 1. The complex 1, it can be also called as NADH coenzyme Q oxidoreductase. Electron transport begins when hydrogen ions and electrons are transferred from NADH having least redox potential to complex 1 using FMN and a series of iron sulfur cluster. Loss of hydrogen from NADH regenerates NAD+, which can be used in glycolysis or TCA cycle. Hydrogen ions and electrons are transferred to the mobile electron carrier CoQ ubiquinone, forming CoQH2. Complex 1 electron transfer. During the electron transfer, H plus are pumped through the complex 1 into the intermembrane space of mitochondria, producing a reservoir of H plus. Hydrogen ion gradient is made. For every 2 electrons that pass from NADH to CoQ, 4 H plus proton are pumped across the mitochondrial membrane, producing a charge separation on opposite sides of the membrane. So NADH plus H plus CoQ gives rise to NAD plus and CoQH2. Complex 2 consists of the enzyme succinate dehydrogenase from the citric acid cycle. Sometimes it is called succinate Q oxidoreductase. In complex 2, CoQ obtains hydrogen and electrons directly from FADH2. This produces CoQH2 and regenerates the oxidized FAD, which can be used in TCA cycle, along with the help of a series of iron-sulfur clusters. 2 electrons are transferred from the mobile carrier CoQH2. These reactions also drive the redox reaction within quinone. These sets of reactions help in transporting the electrons to the third enzyme complex 3. So FADH2 plus CoQ gives rise to FAD and CoQH2. Complex 3, it is called cytochrome BC1 complex or cytochrome C oxidoreductase. It contains cytochrome B, iron sulfur protein, RISC center, 2Fe2S center and cytochrome C. This complex oxidizes ubiquinol and also reduces cytochrome C. The electron is transported via these reactions onto complex 4, accompanied by the release of protons. Now cytochrome C, it contains Fe3 plus and Fe2 plus, which is reduced to Fe2 plus and oxidized to Fe3 plus. It generates energy from electron transfer to pump 4H plus from the matrix into the intermembrane space, increasing the hydrogen ion gradient. So CoQH2 plus 2 cytochrome C Fe3 plus gives rise to CoQ plus 2H plus 2 cytochrome C Fe2 plus, that is in the reduced state from the oxidized state. Complex 4. The complex 4 is sometimes called cytochrome C oxidase. It contains cytochrome C, A and A3. So 4 electrons from cytochrome C are passed to other electron carrier, that is complex 4. Then from complex 4, electrons combined with hydrogen ions and oxygen to form 2 molecules of water. This conversion occurs in the presence of copper ions and drives the oxidation of the reduced cytochrome C. So 4 electron plus 4 proton plus oxygen gives rise to 2H2O. Energy is used to pump H plus from the mitochondrial matrix onto the intermembrane space, further increasing the hydrogen ion gradient. Oxidative phosphorylation. So energy is coupled with the production of ATP in a process called oxidative phosphorylation. In 1978, Peter Mitchell theorized about a chemiosmotic model which links the energy from electron transport to a hydrogen ion gradient that drives the synthesis of ATP. It allows complexes 1, 3 and 4 to act as hydrogen ion pumps, producing a hydrogen ion gradient. It equalizes pH or delta pH, an electrical charge between the matrix and intermembrane space that occurs when H plus must return to the matrix. Oxidative phosphorylation or ATP generation, what does it mean? In the chemiosmotic model, H plus returns to the matrix by passing through a 5th protein complex in the inner membrane called ATP synthase. It is also called complex 5. The flow of proton from intermembrane space through ATP synthase generates energy that is used to synthesize ATP from ADP and PI. This process of oxidative phosphorylation couples the energy from electron transport to the synthesis of ATP. These coupling process can be uncoupled by natural and artificial uncoupler. Sometimes uncoupling is needed in thermogenin, which is present in the mitochondrial membrane of brown adipose tissue. It is the alternative pathway. So here, in spite of ATP generation, thermogenesis occurs. The synthetic uncoupler is 2,4-dinitrophenol. Oxidation of glucose. So cells are able to make ATP via substrate level phosphorylation, that is transferring of phosphate directly from substrate to ADP, and oxidative phosphorylation, that is use of ATP synthase and energy derived from a proton gradient to make ATP. So electron transport and ATP synthesis that I have already told. So reduced coenzyme NADH can give rise to output of 2.5 ATP, and reduced coenzyme FADH2 can give rise to 1.5 ATP according to current research. ATP synthase or F1F0 ATPase is a reversible coupling device that can convert energy of the electrochemical proton gradient into a chemical bond energy or vice versa. The direction depends on the magnitude of the electrochemical proton gradient. In many bacteria that can grow either aerobically or anaerobically, the direction in which the ATP synthase works is routinely reversed when the bacterium runs out of the oxygen. The regulation of electron transport and oxidative phosphorylation. Electron transport is regulated by the availability of ADP, phosphate, inorganic phosphate, oxygen and NADH. When a cell is active and ATP is consumed rapidly, the elevated levels of ADP will activate the synthesis of ATP. The most important known inhibitors of electron transport chains are amytal, rotenone, antimycin A, carbon monoxide, sodium azide and cyanides. Cyanide inhibit complex 4. Amytal, barbiturate and rotenone block the electron transport chain between complex 1 and CoQ. Bacterial electron transport pathways are in general induced, branch and modular. Depending upon the environment, bacteria synthesize different transmembrane complexes as well as produce different electron transport chain. They choose the DNA library for making dehydrogenases, terminal oxidases and terminal reductases. Nitrate, sulfate can be used for anaerobic bacteria as the terminal proton acceptor. Photophosphorylation. In oxidative phosphorylation, electrons are transferred from low energy electron donor, that is NADH, to an acceptor, example oxygen here, through an electron transport chain. But in photophosphorylation, the energy of sunlight is used to create a high energy electron donor and an electron acceptor. Photosynthetic electron transport chains use mobile lipid soluble quinone carriers like phylloquinone and plastoquinone and mobile water soluble carriers like cytochromes, etc. They also contain a proton pump resembles mitochondrial complex 3. Over the thylakoid membrane, they have photosystem 2, B6F cytochrome, photosystem 1, ferrodoxin NADP reductase, as well as they also have ATP synthase for ATP generation. So student, I have discussed about the tricarboxylic acid cycle or citric acid cycle or Krebs cycle along with electron transport system or electron transport chain via oxidative phosphorylation to generate ATP. Thank you.